Hello students, good evening. Happy Saraswati Puja to all of you. May Saraswati Goddess be with you in every time of life and give you all the knowledge, bless you with lots of concentration, a lot of memories, and you can pass that knowledge further. That's my wishes for each one of you who are joining me here. So on this day, I would like to share what I'm reading at the moment. So I'm sharing some very drastic books. So this is one book that I'm going through. It's called, what is this called? Attitude is Everything. If you can dream it, you can do it. So it's just about thinking wisely, thinking good, and believing in something and changing your mindset, having an alternative perspective of one thing. Then you can do many things. Like you can bring a lot of changes. So it's by Michael Lebov. Said, think, act, and talk with enthusiasm, and you will attract positive results. So it's all about think, then you speak, then you act. So that's how your life will be different, like different point of view. So this book is by Jeff Keller. So it's a very small book, around one. I purchased it for 180 rupees, I guess, by HarperCollins. Very small one. So yeah. And second, I hope you all know this one. Robin Sharma 5 a.m. Club. Very must to make your things done, to be focused, to be a good earner, to be motivational speaker, to be a good businessman, to be a good learner, or to be a good family man, to be a good mother, to be a good father, to be a good student. It's all about 5 a.m. Club. If you want to have anything in your life, believe me, if you're starting everything at 5 a.m. in the morning, things will be quite different. You wake up, you do some puja or some meditation, do some yoga or go for a walk or exercise. Then you make your focus, you pen down your things, what you want to achieve in the whole day, and then keep going with that. Another one, that's I got it from the railway track. It's called Chanakya Niti. It's the English version of that. It's very strong book. It's a very strong book. The things that has been written is from the old time when we had this king system, and things are quite hard in this. What they have written, very hard, straightforward, the way Chanakya had wrote. But to some extent, I agree to this book. According to today's world, it's not possible to survive with this Chanakya Niti, because they are really hard and sharp and just straightforward. As somebody is following Chanakya Niti at the moment, they could be successful. Yes, I mean everything has its ups and downs. But me as a person, I it's my belief that I don't want to be judgmental on this. Uh, I it's very harsh for me. Yeah. Then I got this one, 0 to 1. Sorry if I'm not starting. We start mostly by 9 or 10. So I was just going through, so I feel like sharing with you all. This is 0 to 1 by Peter Thiel. He shared his theory, his life, practical life about opening PayPal. If you have might seen that when you're doing online shopping, there's a PayPal thing, yeah. So it just share how Apple and all these big companies from zero they were and how they become number one. So Peter Thiel has shared his own personal tracks. Basically, it's for the economic students who is doing finance. How financial world works while building from 0 to 1. If you want to open your company, if you want to go for something of your own, having maybe your online jewelry shop or any suit, I don't know, watch, stationery, teaching maybe online Excel, There are many things that you have in at the moment, and you can sell it. So this book 0 to 1 will make you go ahead with that. Then there's a Brian Tracy, Get Smart. I haven't read it much yet. It's just some of my friends just gave me this one. If you are like being part of Idofabrica, Anand sir just provide me this. So we are, yeah, I have to see. I don't know what about this. Get Smart. It might be about how you think, how to find the practical solution. Slow thinking versus fast thinking. Oh, family and relationship. So this is also recommended. Go with that. So you also can share with me here what are you reading at the moment. So please make sure you are reading something every day, and you're learning something out of your syllabus, because it's not about what the degree that you're gonna get. It's all about as a person you will get out of your degree, how you shape yourself from your degree. To me as a person after BTech, I was a trash. Yeah, I was not a very like a brilliant human being after my BTech, to be honest. But as gradually masters went, my PhD went, things start to chisel. Things starts to chisel, and you started to shape yourself as a human being. You get to know about yourself, and you are getting more aware about yourself. So that's really important, that who we are, why we are here, and what is the purpose, what is our forte, you know. Okay, Kalpita is saying that Dharma by Amish Tripathi. Shamanism and ancient healing. Abhay, okay. What is shamanism actually? Oh, it's 9 or 10. We should start our class now. Uh-huh. Quite interesting. Inca shamans. Fine, fine. So, in your comments or in the group, keep sharing your books that you're reading, so that everyone could be aware of what we are reading, and we should support each other as a book club, so that we get to know about new things, what's good for us, apart from our course books, Lehinger, Biochemistry. They are also quite important at the moment, because these are the books that will shape your career. So today we were taking Viva, external Vivas of biochemistry and analytical techniques in biotechnology, these two subjects. So in that exams, we were not trying to push students that you have to give me this answer. Yeah, I'm asking you Beer-Lambert's law. What is Beer-Lambert's law? Tell me. But because this makes the students to be down, and you are getting very like nervous, and that's what I saw mostly here in India. It's happening. But our purpose was to teach you something from that even Viva, and if you don't know, we can tell you. Maybe you can read it later. That you come out of it as a good learner, nothing as a bore studies or to take advantage of students, something like that. So on that note, if yesterday at 7:30 our signals went off, so hope so it doesn't went today. It's okay. Um it is fine also today. So if it is so, I will keep my phone with me, so you can message me on the phone, and I will look for it. Maybe I can look from here only when it goes off. Okay, so today we are done with our experiment, with our slides of basics of molecular biology, and we are done with this microscope, cell culture, basics techniques of molecular biology, restriction enzymes, this DNA fingerprinting, RFLP markers, AFLP, RAPDs. Then we went for protein purification, with all different techniques yesterday, and now I'm gonna push you all for gel electrophoresis. So there are many kinds of electrophoresis actually you might be not aware of. So this lecture of today will enlighten you regarding the same. So this will have few videos, not much videos, but let me see, can I have some virtual things for you, because this was not working last time. Hope today it works, because these animations were really good and they were straightforward that we were doing in our lab. No, I don't think so they will work for us. Oh, come on, learn genetics, help us. We want to teach students. Yeah, it will not work. Bad luck. Anyhow, we continue with our lecture then. Electrophoresis. Yeah, so you can see some blurred image here with the chamber attached to the voltage. Two gels are being running left and right with the black spot attached to the black and red one to red, and the voltage is being given here. So electrophoresis techniques, Edu Fabrica. So theory of electrophoresis. So electrophoresis is a physical method of analysis which involves separation of the compounds that are capable of acquiring electric charge and conducting electrodes. So it's a physical method that we know which involves separation of compounds and acquiring the electric charge and the conducting electrodes. And the basic definition is that electrophoresis may be defined as the migration of charged particles to solution under the influence of external electric field. So within the external electric field, you can migrate your charged particles to solutions. So ions that are suspended between two electrodes tend to travel towards electrodes that bear opposite charges. So in this, ions having two electrodes, they travel towards the opposite charge. If they're positive, they will move to negative. If they're negative, they will move to positive end. So there are two types of electrophoresis, that is zone electrophoresis and moving boundary electrophoresis. In the zone electrophoresis, we have four types, that is paper electrophoresis, gel electrophoresis, thin layer electrophoresis, and cellulose acetate electrophoresis. Whereas in the moving boundary electrophoresis, we have four types, that is capillary electrophoresis, isotachophoresis, isoelectric focusing, and immunoelectrophoresis. Yeah, so these are the two different types and having four different types of electrophoresis within them. So we will go each one of them. So at the moment, which is highly being used in the labs, are the like isoelectric focusing and gel electrophoresis. They are being highly used for the reactions and not the other. So zone electrophoresis, so in this it involves the migration of the charged particles on the supporting media. So in this you migrate your charged particles on the supporting media, such as and we use paper, cellulose acetate membrane, starch, gel, polyacrylamide, and components are separated according to the discrete zone on the support media in this. So there are some advantages that they are useful in the biochemical investigations, and small quantity of sample can be analyzed and cost is low and easy to maintain. Whereas the disadvantage is that they are unsuitable for accurate movability and isoelectric point determination. So due to the presence of supporting medium, technical complications are possible. Having electroosmosis, absorption, and molecular savings are introduced into them. So in this, you saturate your media with buffer. Then sample application, you put your sample and you remove the samples with the help of electrophoretic charge. Then you remove the supporting media. In this, we need electrophoretic chamber, electrodes, diffusion barrier, and supporting stabilizing media. So this is how paper electrophoresis looks like. So you have your pencil line here and paper filter here, and you have two glass plates, left and right. You have electrolyte and 45 volt of DC with 5 batteries of 9 VC. So you have within this paper electrophoresis, it could be horizontal as well as vertical. So this is the all apparatus of paper electrophoresis, and on top you can see horizontal, and on the bottom you can see the vertical. So in the horizontal, the main thing is that on the left and right you have positive electrode and negative electrode, and you have buffer solution in them with a filter paper strip attached to it. Then airtight housing is being there so that things could move within this field. And whereas in the vertical thing, everything is same, it's just your filter paper strip is in this triangle form instead of being straight, so they are more on the vertical side. And here we use Wattman number 1 and Wattman number 3 millimeter of paper, and it has a good effect. And in this, separations takes place after 12 to 14 hours. And the advantage is that it is quite economical, easy to use. Certain compounds such as proteins, hydrophilic molecules can be resolved due to the adsorptive and ionogenic properties, results in tailing, distortion of components bands. Now next comes your continuous electrophoresis. In this, what is happening is you have a sample reservoir here, and you have positive and negative charge being there, and electrodes being present here. Then you have filter paper, sample component, and then you can see them zigzag shape. And in the three forms, you will start to have a sample collection. So this is a preparative scale purpose, and predetermined samples at 0.2 millimole through the wall values applied. Silica gel, powered glass, and agar gel can be used in this case, and potential difference of 500 volt is applied, and pure compounds are collected in separate containers, and solvent is evaporated, pure fractions are reused. So after complete electrophoresis, the filter is removed from the apparatus and stained to locate the separated component. In between, I need to check if everything is going fine because my phone is away from me. Okay, all is good. Then comes your gel electrophoresis. In this, separation is done through the molecular sieving technique based on the molecular size of a substance. And gel material act as a molecular sieve here, and gel is a colloid in a solid form with 99% in water. And it is important that support media is electrically neutral. And different types of gels which can be used are agar, agarose, gel, starch, sulfatex, polyacrylamide gels. So porous gels act as a sieve by retarding, or in some cases by completely obstructing the movement of macromolecules while allowing smaller molecules to be migrate separately. So here you can see, cast your gel for making a gel slab, and then you pour your agarose solution on top, and on the left and right you have tape it, and then you put this comb on top. Then in the comb, then you remove this comb afterwards, then you will see some sockets inside as a well. Then you put your sample with the help of micropipette, and then electrophoresis chambers, power supply is done, and then you can see the results afterwards. So in gel electrophoresis is carried out in two methods, that is vertical and horizontal. In the vertical you have starch gel electrophoresis, in the horizontal you have starch electrophoresis. So from negative to positive, your gels are being put up, and you see the results in the computer afterwards. And something about agar and agarose gel. So agar is actually, it's a mixture of polysaccharides extracted from the seaweeds, whereas agarose is a highly purified uncharged polysaccharides derived from agar. So agarose is chemically basic disaccharides repeating units of 3,6 and hydroalgalactose. And agarose gels are fragiles and they are actually hydrocolloids, and they are held together by formation of weak hydrogen and hydrophobic bonds. They are used in electrophoresis. Here you can see in the solid state your agarose looks like this, but in the initial gel, with the increase in temperature of 45 degrees Celsius, they started to make this zigzag helical structure, and then at 100 degrees Celsius they make this final gel structure looks like this. That's how agarose is being made. So something about agarose gel electrophoresis, how it is run. Let's watch it.
This video will take you through the process for loading and running DNA samples on an agarose gel. You will be using a mini subcell to perform agarose gel electrophoresis. The subcell has an electrode wire running across the bottom of each end. This provides for an electric current to pass, which separates the DNA fragments in the samples. First, align the gel so that the wells are closest to the negative or black electrode. DNA is negatively charged and will move from the negative electrode through the gel toward the positive or red electrode. Place the agarose gel into the gel chamber. Add electrophoresis running buffer to the reservoirs at each end of the gel chamber, and keep adding buffer until the wells in the gel are covered by at least 2 millimeters of buffer. Place your samples to be loaded in the correct order according to the lanes they are assigned to be run in. Check your lab protocol for this information. To obtain a DNA sample, slowly depress the plunger on your adjustable micropipette to the first or soft stop. Holding the plunger down, insert the tip of the pipette into the microtube with the DNA sample. Place the tip close to the bottom of the sample, then slowly release the plunger button. When loading the samples, keep the pipette tip perpendicular to the row of wells. This will reduce the risk of accidentally puncturing the wells with the tip. Lower the tip of the pipette until it breaks the surface of the buffer and is located just above or just inside the well. Slowly apply pressure to the plunger button and observe as the sample fills the well. Make certain you stop at the first stop on the pipette, pause, then slowly remove the pipette while keeping your thumb down on the plunger. Once all the samples have been loaded, avoid any bumping or movement of the gel chamber. This might result in the sample spilling into adjacent wells. Place the lid on the gel chamber with the terminals correctly positioned to the matching electrodes on the gel chamber, black to black and red to red. Connect the electrodes to the power supply, again making certain the electrodes match the terminals on the power supply, red to red and black to black. Switch the power supply on. Then set the correct constant voltage for running the samples. See your lab protocol for this information. If a timer is available on your power supply, set the clock for the proper time for your run. Press the start button to begin the flow of current that will separate the DNA fragments. At this point, you should be able to see bubbles coming from the wires at each end of the gel box. Notice that the positive or red electrode has fewer bubbles than the negative or black electrode. In a few minutes, you should see the samples begin to migrate from the wells into the gel. Notice as your DNA samples run, the dye front moves from the negative electrode towards the positive electrode.
So that was the way how it was done. The gel electrophoresis. Now comes polyacrylamide gel electrophoresis, that is PAGE. So it is prepared by polymerizing acrylamide monomers in the presence of methylene bisacrylamide to cross-link the monomers. So your structure of acrylamide, you can see CH2, CHO, and H2. So polyacrylamide gel structures held together by covalent cross links. So polyacrylamide gels are tougher than aqueous gels, and it is thermostable, transparent, strong, and relatively chemically inert. So gels are uncharged in this and prepared in a variety of pore size, and proteins are separated on the basis of charge to mass. Uh, charge to mass. Oopsie. Ratio and molecular size are formally called molecular sieving. And here the gels are quite stable over the wide range of pH and temperature, and different pore size can be formed, and separation speed is good comparatively. Then comes your types of page. So page can be classified according to separations of conditions. So it could be native and denatured, or SDS page. So in the native page, they are run under the non-denaturing condition so that the analyte nature source is maintained. Separation is based upon charge, size, and shape of the macromolecules, and useful for separations or purifications of mixture of proteins. And this is the original mode of electrophoresis. Then in the denatured page or SDS page, separation is based upon the molecular weight of proteins and the common method for determining the molecular weight of proteins. And you can check the protein purity also. So this is, you can see you have anode and there is cathode. And this is the vertical gel. You have large low positive charge and large high positive charge, and you have small high positive charge and small low positive charge. So in this way they will start separating. So in the page procedure, basically your molecules, your protein molecules will get separated according to their mass and charge in this long tube, like we did in this 2D gel electrophoresis. So I will not repeat that part. Okay, then comes your SDS polyacrylamide gel. In this, you have sodium dodecyl sulfate is attached in this technique, quite widely used in biochemistry, forensic, genetics, and molecular biology. So in this, what is SDS is doing? It is being attached to your protein at a constant charge mass ratio. So due to the masking of charges of proteins by large negative charge on SDS binding, the proteins migrates along the gel in order to increasing size or molecular weight. So molecules in solutions in SDS page have net negative charge with wide pH range, and polypeptide chains binds amount of SDS in proportion to its relative molecular mass. Then sodium dodecyl sulfate SDS page, here you can see you have a native protein and which then you heat it and the reductant with SDS. Then this SDS negatively, it will make the protein in the tertiary structure into a linear structure, and this will be a denatured protein with SDS bound to it. So negative charge of SDS will be bound to it, and this will help you to separate your protein, and this will be then coated around it, and according to the size they will get separated, and hence you will see your sample. It's not required starch. And this is also of two types, vertical and horizontal gel electrophoresis. So on the up is the horizontal. Here you put your filter paper strip with starch on the top, and on the other side you have your filter paper connecting strip being attached here. Now before we go ahead, I hope connection is okay. Good evening, bache. You are all getting attached so late. Anyhow, we continue. With SDS-PAGE. I want to show you one video, actually this one. Biomedical, it's by an Italian girl, or I don't know, maybe Spanish, because I can feel her accent, how she's speaking. So this is must video for all of you.
Hello guys, in this video I'm going to speak about SDS-PAGE, sodium DODECYL sulfate polyacrylamide gel electrophoresis. So I will speak about several points. First, what is SDS-PAGE, the sample preparation, the discontinuous gel system, and the separation principle. So at the end of this video, you will get a very good idea about SDS-PAGE. You will understand everything about the principle and the technique. So I will start first talking about what is SDS-PAGE. SDS-PAGE is an electrophoresis technique. It's used for protein separation, and the gel used for SDS is polyacrylamide gel. Acrylamide is an inert material, so it doesn't interact with the proteins, which is a good feature, and it forms a matrix. The pore size of the matrix can be determined by the concentration of the polyacrylamide gel, so it's a good gel. The SDS gel is composed of two layers of the gel. The first is the stacking gel. The second is the separating gel. In the stacking gel, we have the wells in which we apply the protein samples, like here. And then we apply an electrical field to the gel, going from negative on the top to positive on the bottom. The electrical field is applied by a battery. And when the electrical field is applied, the proteins start to migrate from negative to positive, and they are separated in the separating gel according to their, like this, according to their molecular weight. This is the general description, but I'm going to speak about everything in details. First of all, to understand the principle of the SDS-PAGE, we should separation. In the sample, we have the proteins, and we also have what we call the denaturation buffer, or the sample buffer, or the Lamley buffer, which is composed of several substances or materials. And the most important two materials of this is SDS and the beta-mercaptoethanol. So I'm going to speak about them first. SDS is an anionic detergent. It has a polar side and a non-polar side, a polar side and a non-polar side. When SDS is dissolved, it gives a negative ions, and these SDS ions bind to the proteins on the side chain of the amino acids that in the protein. So SDS gives the protein a negative charge. Whatever is the original charge of the protein, even if the protein is originally negative or positive or neutral, the SDS gives the protein negative charge. The SDS also denaturates the protein, so it converts the protein from its tertiary and secondary structure to its primary structure by breaking the bonds that are implicated in the formation of the tertiary and secondary structure. Similarly, beta-mercaptoethanol also has a role in the denaturation of the protein. It's a reducing agent. It reduces the disulfide bond in the protein, so it separates the subunits of a certain protein. So this is the protein, this is the amino acid or the peptide chain of the protein, and then the secondary and tertiary and quaternary structures are formed by non-covalent bonds like the ionic bond, the hydrophobic interaction like van der Waals bonds, the hydrogen bonds, and the disulfide linkage. And these bonds are broken by the SDS and the beta-mercaptoethanol. At the end, we will have the protein as a peptide chain. So the shape of the protein doesn't play a role in the SDS, and also the charge of the protein. So neither the shape nor the charge of the protein plays a role in the SDS page, only the molecular weight of the protein. So this is SDS and beta-mercaptoethanol. We also have the tris hydrochloride, which is a buffer that is of pH 6.8. It also gives the chloride ions. The chloride ions are also important in the mechanism of SDS. I'm going to speak about them. Bromoethanol blue is a staining dye. It's used to give the sample a blue color, so we can detect the sample on the gel, the level of the sample on the gel. Glycerol increases slightly the viscosity of the sample. And also we use distilled water to continue the volume, and we use distilled water because we don't need ions in the sample. Now I'm going to speak about the discontinuous gel system. So we have two layers of the gel, as I said previously, the stacking gel and the separating gel. And they have different pH. So the stacking gel has a pH of 6.8. The separating gel has a pH of 8.8. We also have the two electrodes that have pH of 8.3. There is something called the migration buffer, and the migration buffer contains tris and the glycine. Glycine is also very important in SDS-PAGE, so I'm going to speak about glycine. Glycine is an amino acid, and it's the simplest amino acid because the side chain is only a hydrogen, and it says zwitterion. What does that mean? It means that in acidic pH, so below 2.3, this amino acid is protonated, so it's positively charged. And in basic pH, over 9.6, it's negatively charged because it's deprotonated. Between these two values, which we call pKa1 and pKa2, this amino acid is neutral. And it's totally and exactly neutral in the very middle point between pKa1 and pKa2. So how can we know it? We take the 2.3 plus 9.6 divided by 2. So we get the value of 5.95, which we call the isoelectric point. In the isoelectric point, the molecule is exactly neutral. Above this point, so at pH, let's say pH 6 or pH 6.5, the proteins start to be slightly negative, and the negativity of the protein increases gradually between 5.95 and 9.6. So we should know that in this area, it's slightly negative, and the negativity increases. So let's see. When the pH is in the electrode, the pH is 8.3, and then glycine is negatively charged. As you see here, glycine here at 8.3 is negatively charged. So the glycine will migrate from the negative towards the positive pole because it's negatively charged. But when it enters the stacking gel, in which the pH is 6.8, the negativity of the glycine decreases, and then the mobility of the glycine becomes so slow. So I will speak about each one of these separately. So let's first take the stacking gel. In the stacking gel, the pH is 6.8, and here we have the sample. So in the sample, we have chloride ions. As I told you, we have the tris hydrochloride, which gives chloride ions. We have the protein SDS complex, which is negative, Chloride is also negative. And we have the glycine, which is at pH 6.8, neutral to slightly negative. So they migrate in this order. So first we have the chloride, then we have the protein, then the slowest is the glycine. So as you see here, the proteins are stacked between the chloride and the glycine.
Okay, stream is working fine. That's good.
Glycine. And because of this, we call this the stacking gel, because the proteins are stacked between these two. And when the three molecules reach this line, they start the separating gel. In the separating gel, the pH is 8.8. This is the separating gel. And the pH is 8.8. So the migration takes place in this order. So first we have the chloride, which migrates so fast toward the positive charge. Then we have the glycine, because when the pH is 8.8, the glycine is much more negative. And so it becomes faster than the proteins. And then we have the proteins. So you see here the proteins are left free behind. So the proteins are free behind, there is nothing, and then they are separated freely in the gel. The question is, why do we have this discontinuous gel system? And the answer is, when we apply the sample in the well, the sample is about 3 or 5 millimeters. And then the proteins will not be applied to the gel at the same time. So if the separating gel starts from here, the proteins will be applied to the gel not at the same time. But when the proteins are in the stacking gel, they are stacked together and then they enter to the separating gel at the same time. And they are separated to give the bands, especially like when proteins have the same molecular weight, they should migrate together at the same time to give this band. Now the separation takes place because this gel is a matrix, as I said before. So you can imagine it like this. This is the polyacrylamide matrix. This is the sample. So the sample migrates from negative to positive. And when it starts in the separating gel, the large proteins cannot migrate farther because they stick in the gel earlier. I should say that the stacking gel is a very low concentration of polyacrylamide, and because of this, the proteins do not stick in the stacking gel because it's so diluted. It's not concentrated, so the pore size is so big. But in the separating gel, the separating gel is more concentrated, and so the proteins will stick at a certain point. So the larger proteins stick earlier, but the smaller proteins migrate and stick later. And then we will get different bands of different sizes of the proteins. This was everything I wanted to tell you about SDS-PAGE. I hope you enjoyed this video and you benefit from it. If you have any questions,
so I will share this with you. So this have shown this video of 13 minutes A to Z about your SDS-PAGE. This is all the biochemistry, chemistry, biology, biotechnology, whatever the techniques you want to get attached to it. This one video whole everything about SDS-PAGE in this. Cool. So let's discuss another electrophoresis techniques. Another is now pulse-filled electrophoresis, which is used to separate large DNA molecules by applying to gel matrix an electric field that periodically change directions. So in this periodically change the field of direction with the length of DNA direct to the change at the different rates. So voltage is periodically switched among the 3 directions, one that runs through the central axis of the gel, and 2 that run at the angle of 60 degrees of either side. So here you can see your gel is being run like this, and your angle of your electrophoretic gel pulse could come from any direction. Then after this comes your thin layer electrophoresis. This is done using thin layer of silica, alumina. Advantages are it's a less time consuming and good resolutions. An application is widely used in combined electrophoretic chromatography studies in two-dimensional studies of proteins and nucleic acid hydrolysides. So then there is a cellulose acetate electrophoresis. It includes 2 to 3 acetyl groups per glucose unit, and its absorption capacity is less than that of paper. So it gives sharper bands and provides a good background for staining glycoprotein. The applications are widely used in analysis of clinical and biological protein samples, and it's alternative to protein electrophoresis. And this is moving boundary electrophoresis. The moving boundary method allows the charged species to migrate in a free moving solutions without the supporting media. So in this, you need a U-shaped glass cell of rectangular cross sections with electrodes placed on one of the limbs of the cells. Sample solution is introduced at the bottom, and the apparatus is placed in constant temperature bath at 40 degrees Celsius. And detection is done by measuring refractive index throughout the solution. So you have an anode, you have a cathode, yeah, and you have a protein molecules, and you have ascending boundary here and descending boundary here. So your molecules start to migrate from here till there, and then you see some optical system to be reflected in this. So this is how your moving boundary electrophoresis works. So this is done to check the macromolecular systems and analysis of complex biological mixtures. Then comes your capillary electrophoresis. This is also done at very large level in the proteomics labs. And the main principle behind them is that it includes a charged molecules that will migrate to the opposite poles, separate from each other on the physical characteristics. So capillary electrophoresis is a technique that is done to perform electrophoresis in buffer-filled narrow bore capillaries, normally 25 to 100 millimolar in internal diameter. So high voltage is applied in this. Capillaries are typical of 50 micromolar inner diameter. And this is how your capillary electrophoresis looks like. So you have a cathode on one side, anode on another. And then you provide a high voltage injection. Potential is applied. And then combinations of ionic attraction of electroosmotic flow is done. Then pressure injections. Pressure difference is used to drive the samples into capillary by applying vacuum into it. Then when potential difference is applied, net migration occurs in the direction of cathode, and even substances with net charge migrate in the direction of cathode due to the phenomena called electroosmotic flow. And the neutral molecule moves at the same speed as the EOF. When it comes, but basically, students, you have to, in all these techniques, they are actually theoretical part. Mostly they are not used in the labs, but whosoever wants to study from the examination point of view, yeah, from the examination point of view, they are quite important. Yeah, if you are like having a technique exam, so all these techniques could be asked. So you just should remember the diagram and what is the basics principle behind it. But you should remember the SDS-PAGE technique that I have shared. That is quite important in all. So capillary, then electroosmotic flow, and in this you have a glass capillary, and then you give an electroosmotic flow to that, and surface of the silicate glass capillary contains negatively charged functions groups that attract positively charged counter ions, and positively charged ions move faster and negatively charged ions move slower. So like this, you have a buffer electrode, high voltage given to it, a capillary tube is there, and then at the end you can check your data acquisition in between. And this is a isotachophoresis. This is a development depend upon the development of potential gradient. So based on the principle of moving boundary electrophoresis, so leading electrolyte with a higher mobility than analytes and trailing electrolyte with lower mobility are used. Solution in which separations takes place is normally in aqueous medium, which contains sucrose to provide a higher density to the solution. So where the separation by isoelectric focusing depends upon the existence of pH gradient in the system, the technique of isotachophoresis depends on the development of potential gradient. So in this example, you can see that there are different three particles classes with different charges are being separated and pre-concentrated by the electrophoresis. So after the separation conducted, particles moves at constant speeds, A, B, C. In 10 seconds, they move at different concentration. So you have an electric field being moved from your leading electrolytes, electromagnet, and then you have different results. Then comes your immunoelectrophoresis. This is basically deals with your antibodies that are produced by immune system in response to foreign macromolecules. So each antibody binds specifically to one feature or one macromolecule, and this allows the use of antibodies for detections and quantitation of specific proteins in a complex mixture. So antibody is placed in rough cut parallel to the direction of the electrophoresis, and they run the electrophoresis as results precipitate arcs due to the antibody antigen complex formation. So you have antibody and you have antigen, and they make these unpaired arcs. On the basis of that, we can differentiate between your samples. Then isoelectrofocusing that we did yesterday. This is 2D gel. You run your samples on the 2D electrophoresis pH gradient. They will find the PI and then they will not move forward. So that was about, and then comes your 2D gel after isoelectrofocusing. That's we also did yesterday. So that's it. So that was, so there's some paramecium parlor. So this is the guy in the great costume party. So that was the last year where DNA invited the restriction enzyme to their Helen party. So this is the restriction enzymes who wanna cut this DNA. So I will end this lecture of electrophoresis on this joke of paramecium electro and restriction enzyme here, and will share with you. And also I have one protocol for you, so you can go through it. Gross gel electrophoresis protocol. So interesting thing for you. Okay then, goodbye, take care, wish you all good evening. And yeah, do the best, think the best, make the best, motivate others, whosoever is around you. Keep spreading the light that you have in, and let the other lights be grown with you. Never ever let the other light to be diminished under your influence. Thank you very much on that note. Bye bye. Good night.